Genetics lesson 2.5, so solving genetic problems using Punnett squares. So what are Punnett squares? So in the last few lessons, we talked about words like homozygous, heterozygous, dominant, and recessive alleles, but what do those actually mean in real life scenarios? So Punnett squares help us determine what the kids or the results or the next generation of the cross of the parents will be. So just like how Mendel determined the F1 generations. So essentially it's taking two things, crossing them together and see what it will give us. And the Punnett squares are a really cool tool that people like doctors and genetics and fertility specialists use to determine the traits of the parents and what that will give you for the kids. So we're going to learn how to do those. So before we start, we should probably learn a bit of the terminology for Punnett squares. So like we said before, the genotype is a combination of two alleles, and the phenotype is how we actually express those alleles. So I've written out these two words sort of 2 times so I can give you an idea of what they actually mean. So homozygous dominant in our case is going to be written like this: big P, big P. Now it can be any letter of the alphabet as long as they are both capitals. Like I could do big T, or big A, big A. It doesn't matter as long as they both are capitals to show that it is dominant. Because remember, what does homozygous mean? Homozygous means the same. So they both have to be capital letters. The next one is homozygous, so again, same, but this time recessive. Recessive means sort of like lowercase, so we would do little p to show that it was recessive. But like the homozygous dominant, they both have to be little p because they are homozygous, meaning the same. Heterozygous, on the other hand, means different, so it's sort of a combination of the two. Since we picked the big P to be dominant, it would be big P, little p, so a mix of the two. So if I reword this a little bit, the dominant allele is big P, so that means that heterozygous has to have the big P, and also a little p from the recessive. The big P is dominant. It's telling the little p what to do because it's bigger than the little p. And in the other ones, this full thing is dominant and this full thing is recessive. This is also dominant if we think about it, because this big P has control over this one. It's telling this whole thing to be dominant, just like this one, because the recessive does not have enough power over the dominant. I wrote it out again at the bottom here because the first time we wrote it was the genotype. The phenotype is actually how we talk about it. So if I made big P represent purple, and I made little p represent white, big P, or purple, would obviously give us purple. Little p, or white, would give us white as the result. And then big P, little p for heterozygous, since the big P is telling the little p what to do and big P is purple, it's going to be purple. So now that we have all that terminology covered, we can actually start learning how to do the Punnett squares. So you should make a little note of these steps so the questions are easy for you to follow. So the first one is state our variables. Just like we did in the other question, we made the big P represent the purple and the little p represent the white. Then we show the actual cross. We make the Punnett square, which I'm going to show you how to do in a second. We write the genotypic and phenotypic ratios. So remember, for genotypic, we are going to use the big P, little p, and big P, little p. For phenotypic, we're actually going to use the actual words. So we're going to use like purple, white, stuff like that. And then we answer the full question. So sometimes it could ask you to calculate like it in actual numbers. So you would divide it by a percentage. But I promise you'll learn how to do those harder questions later. But let's start off with some easy ones. So how do we make a Punnett square? So if I chose to cross a dominant purple plant that was homozygous, because they're same, with a homozygous recessive, because they're little p, white plant, cross them, this is how I would draw the square. So you just draw a sort of box like that, or T shape with slightly longer sides, and you would write one of them on this side, so like that, and the other one on the top like this. Doesn't matter, I could put these the purple ones on this side and the red ones up there, it doesn't matter as long as you write them. Then you cross them together. So when we combine these two like that, we get a big P and a little p. We're going to do the same thing. Cross those two up there like that. We get a big P and a little p. And we do the same thing for the bottom part too. So we cross those two up there. We get a big P and a little p. And we cross these ones like that as well. And we get a big P and a little p. So that means that all of these guys' children up here are going to be heterozygous. And they're going to be purple because the dominant allele is purple. So now let's actually start doing a real question, which is what we call a monohybrid, or like the normal way. So in mice, G for gray, the coat color is dominant to G, little g, black coat. So it already tells us that the big G is dominant and the little g is recessive. The little g individuals are black, and 2 gray mice are mated to produce 9 gray mice and 2 black mice. So we have to explain the results. So if we break this down really far, we get that big G equals gray and that little g equals black. Super simple. It tells us that 2 gray mice are mated to produce 2 gray 9 mice and 2 black. So that means that we have to cross a big G and a little g times a big G and a little g. So we set up our Punnett square. So it doesn't matter where we place these. We get G, G, big G, little g, big G, little g, and little g, little g. So we can say that our phenotypic ratio, sometimes I just like to write a little p to represent that. Let's count how many of them are gray. Well, big G equals gray. So this one is gray, and there's no more big G. But we know that the big G is dominant to the little g. I see that this one has a big G, so I'm going to circle it, because that means that it's still gray, because the big G is dominant to the little g. This one is also still gray, because the big G is dominant to the little g. This one, however, is not gray, because it's 2 of the recessive alleles, which means that it is black. So for our phenotypic ratio, 3 of the mice will be gray and 1 of them will be black. And it's important that you write the names above. So this is going to be a little bit harder one, especially because it is a dihybrid. And you'll see that the answer for all dihybrid questions are actually the same phenotypic ratios, but we'll get into that. So in pea plants, tall or big T is dominant to dwarf little t, and round seeds, big R, is dominant to wrinkled seeds, little r. Use a Punnett square to determine the fraction of the offspring produced in each of the following crosses you would expect to be tall with, in each of the following crosses, you would expect to be tall with wrinkled seeds. Okay, so let's just start off by writing out our stuff in our steps. So we have big T equals tall, little t equals short. Then we have big R, which is round, and little r, which is wrinkled. And now we just have to do a cross between the two. So we have a tall, wrinkled seed. And now we have to understand the big T is dominant to the little t, so it's still tall. And a big R, which is round, is still dominant to the little r wrinkled seed. So we have to cross the two. So now we can do our Punnett square, and this Punnett square takes super long. So we have big T, big R, and you just sort of put them together as how their capital letters would go together. So then we have a big T, little r, so sort of like the combinations they could be. But I promise you won't have to do this chart ever again if your teacher asks you for a dihybrid problem, because they are the same answer every single time. And then little t, little r. So this one is going to break down to give us big T, little big R, big T, big R, little r, big T, little t, big R. And I'm just doing the same thing as I was last time, just crossing them together with the top ones. You might start to run out of room, but try to keep it as neat as possible so you can figure out the end results. Just extend that a little bit more. Okay, so now you got to count them up to see how many are tall and wrinkled. So I count this one, it's big T, big R, which we can look at our list. That is, in fact, tall and wrinkled. Same with this one, same with this one, same with this one. And remember, even though there is some little r's and little t's in there, the big T and the big R are dominant over them. I see this one, I see this one, this one, this one, and this one. So I count 9 of those. Next, we got to count how many are, these were tall and round. So we can write tall and round there. Now we have to find which ones are tall and wrinkled. I count for tall and wrinkled. I have this one, I have this far one, and I have this one. So I count 3 for tall and wrinkled. For short and round, I count, let's see, 1, 2, 3. So 3 again. And then the last one has to be short and wrinkled. Then I count 1. So our phenotypic ratio is going to be 9 to 3 to 3 to 1. And you will never have to do that long chart again because every single dihybrid problem will give you a phenotypic ratio of 9, 3, 3, 1. And that's all for Punnett squares.